I'm Catherine Lambrecht with the Illinois Mycological Association. So we're going to turn over the conversation now to Todd Widhelm. It's his first year as a fungal collections manager. But I also want to highlight that the relationship the Illinois Mycological Association has with the Field Museum is quite an important one, especially when it comes to things like getting our we make our contributions, of course, to the herbarium, but at the same time, the relationship also helps us to get our permits and such. And, Greg Mueller, before he went to the Botanic Gardens, was with the Field Museum. Patrick has been with the Field Museum. So it's a long-term relationship. So this is just the newest one to encounter, which we really appreciate. So Todd, I'm going to turn it over to you to introduce yourself further.
Great. Thanks, Catherine. Well, I feel real honored to be here. It's one of the very few talks that I've actually been invited to. I've always gone to conferences and presented, but to be invited is an honor. So thank you for that. And I'm looking forward to continuing collaborating. And for me, being a lichenologist, I don't have a lot of mushroom identification skills, which I really need to work on. And so I apologize right off the bat. There's not a lot of Basidiomycete fungi in this talk. It's very lichen heavy, but lichens are fungi too, and I just want to kind of explain what I've been doing in this first year. So I'm going to start sharing my screen. Let's see here, share, and I'll pop this up. So when I was when I was thinking about, what am I going to talk about? I was like, well, I could talk about the collection management. I could talk about my research. Basically what happened was like, well, I'll just give an annual review of my first year. And so sorry, again, right off the bat, it might seem kind of all over the place, but I tried to do my best to give it some organization. So I just want to introduce myself. As you might have heard, if you were in at the beginning, I'm from Nebraska. I identify as a Nebraskan, and I did my undergrad at the University of Nebraska at Omaha, and that changed my life, really. I used to go hiking in the woods in my hometown and saw all these weird things on the trees, and I didn't really know what they were. And then when I took botany and then eventually lichenology with Dr. Bob Egan, I learned what all this stuff was, and I just became really extremely interested in lichens. He took us on field trips to the Rockies and to the Ozarks, and then I also continued doing my master's degree there. And then after that, I came here to Chicago at the University of Illinois at Chicago at UIC. And I did my master's with Thorsten Lumbsch, who's the curator of lichens at the Field Museum, and Roby Mason-Gamer at UIC. So I did field trips. My work was in Australia, New Zealand, and Chile. And I worked on lichen evolution for my systematics. But going from PhD, I thought like when I finish my PhD, my life's going to be so much easier. But really, everything got way more complicated, more complex, I guess. And So now I'm at the Field Museum as collection manager, and I basically, the way that this talk's going to go is what I see is like the 4 broad areas that my work encompasses: collection management, education and outreach, field work, and research. So we'll first start off with the collection management part of my job. And being a collection manager has a lot of different tasks. There's identifying specimens, handling loans and exchanges with other institutions, writing grants, digitizing specimens, and then physically processing specimens and getting them organized in our collection. And the place where everything lives is in the Fungal Herbarium at the Field Museum. We have over 300,000 specimens with a huge collection of Basidiomycete fungi. A lot of those we have a huge collection of types from Singer, Greg Mueller, Patrick Leacock, Sabine Hundorf, a lot of people that names that you might be familiar with. Their specimens are in our collection. We also have a very large lichenized Ascomycete collection that's been accumulated. That there's a lot of historic stuff, and there's a lot of stuff that's recently collected due to research from Thorsten Lumbsch and all of the students that work with him, that even like from me, and also Robert Luking, who I replaced as collection manager there. And there's a lot of work to do, I found out. And this is the 6th largest fungal collection in the US. We have other laboratory spaces, the Fungal Culture Lab, where it's been active. Sabine Hundorf has been in there culturing lichen fungi for genomics. With lichens, you have the symbiotic relationship, and it's really useful to have a pure fungal culture of the lichen fungus to do genomic work with. She's been a great help in the Fungal Culture Lab. We also have a thin layer chromatography lab. You can see here in this fume hood here, and then here's a plate showing some lichens that I spotted on, and thin layer chromatography is for certain lichens, you have to do this to identify the species. You need to do thin layer chromatography. And then we also have a microscopy lab that has cameras on microscopes hooked to computers, and we can make nice images of microscopic pictures. And then we have a digitization lab as well, where we're taking images of the labels of specimens and converting those into digital format, entering those into our database. So they're available online, and you can actually go and search any of our records in the botanical collections at this link. So I was preparing a grant and just trying to figure out how many lichens were collected recently. And just within the last 3 years before 2020, which I don't think much is going to happen in 2020, but we had 5,000 lichen specimens collected. And also many Basidiomycete fungi were collected too. So we have all of this going on, and on top of that, there's just accumulating work with DNA sequencing of fungi going on. And these charts here, I pulled out of this Kew Gardens report on fungi from 2018. So on the left, we have the cumulative number of fungal families published by decade. You can see from 1800 to 2010, it's just gone up almost at an exponential rate. And then on the other side, on the right, we have a bunch of, we have 4 different genera of fungus that have since basically around 2010, that's when DNA sequencing technology became really more available to everybody. And you can see the number of species described in these genera have increased dramatically. And so in 2017, there were over 2,000 species of fungi described. And
Okay.
that's why our collections are super important. Fungal collections all around the world, they keep type specimens for all these new species. And we're also finding new things as we keep going through all of these specimens. And I can't do all this on my own. This requires an entire team of people that help with identification, with processing, with databasing. So I have a lot of volunteers that are working for me. And Wyatt Gaswick is my collections assistant. He's awesome and helping me on the fungal end of things. That's kind of how I have this divided, because there's kind of a lichen side and a fungal basidiomycete side of the herbarium. And Patrick Leacock is for helping with identifications. He's just great. I need that help on the basidiomycete side of things. So with right now, what I'm focusing on is a lichen digitization project. And right now there's about 4 different states that the specimens are in. There's about over 33,000 specimens that we're dealing with here. Currently, we're focusing on Protocol 1, where the specimens are in good condition and they're already in herbarium quality packets, but they still have never been entered into the database. So right now, there's updating the taxonomic database in our EMU database, and then the data entry for those, and then taking images of those labels and then putting them onto the shelves. And then we have all these other different specimens in different states. Some are already are just still in their collection bags, sitting there with only some writing on the collection bag. Others have been kind of processed into temporary packets, and some have been identified partially, but they still need a lot of work. So this is an ongoing project that we're working on at the moment. And since we've had this shutdown, I've had to go in and grab specimens. So I actually went in 2 weeks ago, grabbed a bunch of specimens, processing supplies, computers, and then I drove them out to a couple volunteers to help me with so they can keep helping me do the digitization process. So thanks to Betsy Shepard for processing Madagascar lichens. This is a picture from her home. She has a beautiful view that looks out in her backyard, and she's working on the Madagascar collections that were recently made. And this is hands-on work where she's actually pulling the specimen out of a bag, folding packets, and then putting them on a card with cotton and labeling them. So they're getting organized so I can identify them. So I also want to say thanks to Robert Saul. He's been, I brought a bunch of specimens that from that Protocol 1 that I just mentioned. He is working on updating the taxonomy, because these are old specimens. They need to be annotated, and some of the names are out of date. So he's working on updating that and then databasing those. And he's also helping Wyatt with a georeferencing project. And that leads me to Wyatt, where I have so many thanks because there's so many people working on things and helping with the collection. And Wyatt's been doing a big microfungi digitization project and leading a georeferencing with a bunch of volunteers from the museum, which Robert's one of them. Now I'm jumping to the education and outreach part of the presentation. And we at the museum, when things are normal and running well without COVID going around, we have these we dig bio in the collections club. And so these are citizen science events that have that really focus on doing data entry of specimens and physical processing. And you can kind of see some people here. We get people from we get lots of young students and people that are people of all ages. And We Dig Bio is annual in October, I think, and it's a 4-day event of just digitizing specimens. And then Collections Club was invented by Matt von Conrat at the Field Museum, who's the collection manager he's the head of botanical collections and the head and collection manager of bryophytes. And every quarter we have this Collections Club, which is a 2-day event over the weekend, where it's modeled right after the We Dig Bio program. So if you're interested in this, please reach out to me. I'll share my email later. And the Field Museum just always gives me a place to teach. Even though I'm not at a university, I still get to do I've done the field spotlight where I've talked to people. I've actually met people that became my volunteers there. Then we have the Meet a Scientist, which is an actual exhibit at the museum where we can set up for a day, for a couple hours, and just have people come in and talk to us. And I'll teach about lichens. Do a similar thing at Members Night and the Dozin with the Dinos event. And then just doing public speaking is another big part of my job. I try to attend as many conferences as possible. Where, like, I was supposed to go to a conference in August with the International Lichen Association, and that got canceled. So, but yeah, last year I was in Arizona for the botany conference, and then I was in Quito, Ecuador, and then I was invited to give a talk at the University of Antananarivo in Madagascar. There's a lot of public speaking. But now I'm moving on to the fieldwork aspect of the job, and maybe my it is the best part because you get to actually go and see the forests whatever ecosystem I'm collecting in a different part of the world, and it's always a great experience. And so this year I went to Madagascar to collect lichens and to teach. So a lot of times field work and education is mixed together. So just to orient you, where Madagascar is right off the east coast of Africa. It's the second largest island country, and it's a biodiversity hotspot. Over 90% of the wildlife there isn't found anywhere in the world. And it's got a huge population for the size. And Madagascar is roughly the size of California. And there's 26 million people living there, which you can imagine that it's putting a huge stress on all the ecosystems there. So I was invited by Steve Goodman to go out. So Steve is a Field Museum employee. He's been working for the Field Museum since the 80s, but he's lived in Madagascar the whole time. He started the association Vahatra, and the goal of Vahatra is to understand the biodiversity of Madagascar and to promote conservation and to educate the people there to be able to study, you know, what is right outside in such a unique place. And so it was a real honor to go there and work with Steve. And he usually works with animals. And so he was like trying to get Thorsten to go there, and Thorsten's vice president of the Field Museum, and Thorsten Lumbsch, and he just so busy. So then that allowed me to go, and so it was a great honor. So the first place that we went to was Ambohitantely, which is in the central highlands. It's about a 2.5-hour drive from Tananarivo. They call it Tana there. And it has montane forests, grasslands, and savannas. And they were having a training program for professionals like park rangers, conservation scientists from Madagascar, the Comoros Islands, Mauritius, and Reunion. And here's the team, here's the whole group on the last day. And we stayed in tents for 10 days. My tent is there on the right. We had a whole tent city going on. And the center of activity was this dining hall. And we ate a lot of, in Madagascar, you eat rice with every meal. And Steve asked me, the first day I got there, he's like, do you take a Malagasy portion of rice or a regular portion of rice? And I learned that, like, what I showed you on this plate here is about a quarter of what a Malagasy guy would eat. But we were eating a lot of zebu, which is the beef there, and lots of beans and vegetables. The food was actually really great. Hot cooked meals every day. We had lectures every day. I gave a lecture on lichens. Everybody who took a turn. So what was going on at this workshop was that there were people studying plants, people studying birds, and the attendees of the conference would actually switch groups every 2 days. So I would have a group for 2 days of like 2 to 4 people, and we'd have a lecture, and then we'd go out and then I would teach them lichen collecting techniques. So here we are out collecting lichens in the savanna ecosystem, in the grassland ecosystem, in the montane forests, and out again in the grasslands. And then once we would go out all in the morning and collect, we'd bring everything back in the afternoon. After lunch, we would start identifying them and then databasing. And just to give you, now I have a bunch of pictures of lichens from montane forests. We have big leafy Pseudocyphalaria species, many crustose lichens, this heterodermias and leucodermias, many parmotremas and Physiaceae, cococarpias, heterodermias. It was just beautiful here. I really enjoyed this spot. And when you go out into the savannas, you get a totally different community of lichens. And so there's more light, and you get more usneas, more crustose species, Lobariaceae. And then out in the grassland, you start to have rock outcrops where you have lichens growing out on the rocks, and even things growing on the soil like this Diploschisthes. So after Ambohitantely, we packed up, went back to Tana, had a couple days off, and then we got back in the cars, loaded everything back up, and drove to Ankarana, which is a 2-day drive. It's only 1,000 kilometers, which on well-paved roads, you could drive it in a day. But in Madagascar, the roads are really rough. And we had to sleep one night halfway there and continue driving. It wasn't pleasant, but it was worth it. And this time we had a group of students from the University of Diego, which is up in the northern part of Madagascar. And again, same style. They were rotating around, learning different organisms and the field techniques involved. And lichens was one of the stops. And one thing about Ankarana is it's well known for its singy formations, which are these limestone rocks that came out millions of years ago and then have been eroded by rain for a long time. So they become really sharp and jagged. If you fall on them, it would really hurt. Here's some more of the singy, and this is actually from National Geographic, showing how these form over time. So if you want this, send me a message and I'll send this to you. It's actually really cool. And I have way too many pictures of the singy because it's just so beautiful. We stayed at this camp called Campment des Princes, and you can see how we're all set up. And fortunately, we had a water tower that was put in only a few years ago, and it was actually donated by 2 anonymous women from Chicago who greatly enjoyed their visit to Ankarana. And so thanks to this, we actually had some running water so we could have a big group out there. And so just to give you an idea, that this was our, again, our lecture hall, lunch place, dinner place. We have our bathrooms here. And then where our tents were set up, you can see mine was set up. The roof is gone where I set my tent up, and then where other people have that they don't even have. Some people did have roofs, and it was really hot and humid here. I was trying to constantly dry out all of my clothes. It was like sleeping in a sauna every day. It was rough, but when you're out collecting, it's totally worth it. So we would go to this bridge. Everybody wanted to see the bridge because it was an excellent photo opportunity. And while we were collecting lichens, we would have to go. Everybody was like, "Can we go to the bridge?" And so I got a photo of almost everybody there. And then we went to there was a cave there, which didn't have much for fungi or lichens, but it did have a lot of bats. It was an interesting place to visit. So again, we did a lot of lichen collecting, and these guys just found it fascinating. It really, in Madagascar, a lot of the focus is on animals and plants, and really lichens and fungi in general are just totally overlooked. And just to be there and to show people like this is a lichen, and just like put it in the mind, and I think that everybody really got a lot out of it. And just these are just a bunch of lichen photos, and then just some of the lichens that were there. Yeah, and then also the wildlife, there's terrestrial crabs. This crab here actually has a millipede in its claw. There's these geckos everywhere, many types of geckos actually, and different birds. And every time I was out in the field with these guys, they would also collect snakes. And then they're trying to get me to hold them. And I'm like, no, thank you. That's why I study lichens. And of course, there's lemurs. And they would actually come up while we were eating and beg for food. They are not shy. In total, in Madagascar, during that time, 600 specimens were collected mostly by the students attending the workshops. I was just kind of standing there telling them like, yeah, let's collect that. And then, but what we have now are 600 new specimens to the Field Museum, and then the duplicates are at the University of Antananarivo, which is the first lichen collection that has been there. So it's a first start to a reference collection where maybe people in Madagascar who have way more access all year round can start going and collecting and learning what the lichen flora is. Because I'm sure just like how there's 90% of life in Madagascar isn't found anywhere else, there's probably lots of lichens that are endemic to Madagascar too. So there's a lot of work that needs to be done there. And we're hoping to be able to get grants to do more research down there and teach more workshops. So now I'm moving on to the last part of the talk about the research, which is what got me to the museum in the first place. So I actually met Thorsten Thorsten Lumbsch here in the middle, at a conference, and I was like, I want to do my PhD. Are you taking students? And he said, yeah, apply to UIC or U of C. And I got into UIC, and I basically was working with him at the Field Museum, studying lichen evolution. And he's produced 8 PhD students. He's had 7 postdocs, and there's been so many species described. It's really an honor to work with him. And we're really interested in studying biological diversity. And the tree of life is so big, you almost have to focus on something. And so here's the fungal branch, which is probably really underrepresented in this cartoon tree of life. But that's where we focus on. And in my dissertation, I focus on this family Lobariaceae, which are these foliose macrolichens, over 400 species, and they associate with green algae and with cyanobacteria. That's how you can see some, like this one here, actually is associating with both at the same time. It's what we call a photosymbiodeme, where it has a cyanobacterial association here, and then one lobe actually has green algae. And the project I'm gonna wrap things up with is about Pseudocyphalaria glabra. It has this disjunct distribution. It occurs in Australia, New Zealand, and in Southern South America. And I just thought this was like such a nice system to study whether vicariance, the breaking apart of continents and drifting away, was the cause of this distribution, or if they're dispersing, are they jumping over the oceans? So in a previous study, studying all Lobariaceae, we find that Pseudocyphalaria glabra is within 5 million years old. So you can look over here on this map showing the breakpoints in millions of years ago of the continents of Gondwana. And so 5 million years is relatively recent. Actually, the continents were almost in the same position that they are now today when glabra became when glabra evolved and became a species. So we can probably rule out the vicariance hypothesis. And we're really dealing with long distance dispersal here. And with things with such small spores, it might not be that big of a surprise these days. But there's this dominant weather pattern called the west wind drift. And I always like to show this, this diagram here. This is the path of a weather balloon that was released from New Zealand. And it circled for 102 days at 12,000 meters. And it did 8 circuits before it fell down in South America. So it's not hard to imagine that microscopic spores could do the same thing. And we actually see with a lot of other plants, like ferns, things that produce and other fungi, there's a similar pattern. There's also birds that could be carrying these spores. And if you look at albatross, this is just a map from Wikipedia, but just showing the density of where the albatross is. And glabra occurs here in South America, Tasmania, and in New Zealand. And these birds, these species here of these petrels, they all occur in the same places too, and are migrating in between. So they could be a potential vector. So I had 2 hypotheses. If these large bodies of water can isolate the populations, we should see genetically distinct populations. But with these strong wind patterns and migratory birds, we should also see long distance dispersal. So I went first to Australia to go collect lichens, and it wasn't even for this project. This project was an accident because I was going to Australia to collect for a different project, and I was hardly finding anything, but this weed, which is Pseudocyphalaria glabra, is everywhere. And so I couldn't help but collect it in Tasmania and in the mainland. And that's where I started thinking, like, maybe this is actually a more interesting project. And then that led me to go to New Zealand to collect in the North and South Island there. And you can get an idea of what the habitat is in these beach forests or temperate rainforests. They get lots of precipitation. It's also in the Valdivian rainforests of Chile. Here it is dry. It's actually a really beautiful lichen. Here it is more hydrated. There's one by my head. You can see they get about the size of my head on the tree. And when you look at it up close, it's really easy to identify in the field. It has these brick red apothecia. It has these finger-like projections that we call phyllidea. And then when you look at the underside, it's like this chocolatey brown with these white dots. So those characters, you can pretty much know that it's glabra. And I was trying to collect about 20 individuals per site if I could on different trees. So I knew they were different individuals. And then I brought them back, extracted their DNA, and then I did this method of DNA sequencing called RAD-seq, which is restriction site associated DNA. And it doesn't sequence the entire genome. It only sequences certain parts of the genome. So if this is the whole genome, we only sequence 3, what we call RAD loci. But I was getting thousands of RAD loci per individual. And what we're looking for in each one of these are single nucleotide polymorphism data, or SNP data. And to get an idea of what a SNP is, we have 3 humans here. They all have the same gene, but at the SNP, they're different. So these data here are used to infer the population structure, and we can make these plots, and we can make phylogenetic trees to understand the population structure. So the other thing is, if you're working with lichens, you're working with a symbiotic system. So when you extract DNA, you're not only getting the fungal DNA, but you're getting the algae, the cyanobacteria, and all the other bacteria that live on them. Lichens are like sponges. And so we had a microbiont genome sequenced that we were able to map all of our reads to and filter out all the other DNA from other organisms to be able to study just the fungus. And the first thing I did was generate this tree. And it's a circular tree here. And I just want to orient you to the colors. So Chile is in this gold yellow color. Victoria is like brick red. Tasmania is aqua. North Island, New Zealand is dark green. And then this tan color is the southern island of New Zealand. And looking at this tree, it's actually doesn't make complete sense. You see some nice clustering based on the geography, like this big clade from Chile, this Australian clade here, this New Zealand clade here, but then you have these other ones where it's just not, it's hard to know what's going on by looking at a tree. So we have other methods like principal components analysis, or PCA, where now you can see here each one of these dots represents one individual. And think about this as genomic space. The closer the dots are together, the more similar their genomes are. And here we can actually start to see some different clusters here. A cluster that's associated with Australia, a cluster that's associated with Chile, and then this one with New Zealand. And there's some outliers from each. It's not perfect, but it's pretty, this is where the picture becomes a little more clear, where we're seeing genetic differentiation. We can actually see that the Pacific Ocean and the Tasman Sea are acting as barriers that are isolating these populations and allowing them to become unique genetic clusters, which supports the first hypothesis that these large bodies of water will isolate these populations. But just looking at them with my own idea of what the populations are based on geography isn't the best way to do a thorough analysis. So another way of doing it is to let the genetics do all the work and let an algorithm tell me how many clusters there are, and then to identify, and then to see where are those clusters located. So I did this k-means clustering algorithm, and I found that 5 had the best fit. So the lower the BIC number on this y-axis, the better. So 5 was the best, but you can see that 4 and 6 are not too different. So I actually just decided to look at 3 through 6, and that's what's going to be in this next picture here. We have 3 on the top and going down to 6 clusters. So assuming there's 3 populations or 4 populations and so on. Each one of these bars represents an individual, and the color represents which genetic cluster they are assigned to. We also have them sorted by their where they came from. So as you add more populations, you get more detail, and you can see that around cluster 4, that's where, or when you have a K of 4, you see that Australia is unique, and Chile is unique, and New Zealand has like 2 different clusters. It has more genetic diversity. But when you look at this cluster 4, you can see that group 1, this purple color, is only found in Chile. Group 4, which is only found in Australia, but groups 2 and 3, these colors here, are also found in Chile and also found in Australia. So it seems like we're having a long distance dispersal out of New Zealand to these other places, which also supports hypothesis 2. The strong wind patterns or migratory birds are causing these long distance dispersal events to happen. And we're actually able to capture it in the data. So which of these vectors are causing it? I'm still not sure. If it's the west wind drift, I would expect a west to east pattern of dispersal, but we don't see a lot of Australian populations in New Zealand. And with migratory birds, we could have dispersal in each direction. There could also be floating vegetation rafts that are carrying the lichen around. Right now, I'm still working on that. All could be playing a role. So I'm working on modeling these dispersal scenarios, and I also want to look into the different symbionts because I still have their DNA too, and I can use the same methods to pull out the algal DNA and study that, and to pull out the cyanobacteria that live in there and study them. So that's kind of what I'm looking into more in the future. So yeah, well, that's pretty much all that I have today. I want to thank you. And if there is a little bit of time, I just wanted to show a few pictures from this weekend. So this is my first time posting onto iNaturalist. And so I actually my wife and we went to Backbone State Park in Iowa. And it's about a 4-hour drive from Chicago. And it's actually a really beautiful place. I had never heard about it before. And we just decided, like, we'd been stuck in the house for so long, and we needed to get out and do something. So we went on a camping trip, and it has these amazing dolomite cliffs. I never thought this was in Iowa, but there's not a huge lichen diversity there, but there was some interesting dermatocarpons on the rocks. And but I really focused on taking more pictures of the fungi. So we have the Ascomycetes in the genus Pizziza, the orange peel fungus was there, dryad saddle, the golden oyster mushroom, which we were really happy to find, and the genus Mycena, the orange Mycena, and Tremella was there. Also false turkey tail, which is everywhere, and the Neophalvalus. Thanks, Patrick, for the identifications on iNaturalist. And also some Myxomycetes were there too. So these I posted all on iNaturalist, and you can see where they were. And if you're ever at the field, whenever the museum and everything comes back to normal and you're there, you can also just come and look at the lichens growing on the museum. We have these, all this stuff that they clean off the museum every once in a while, which I call lichen genocide. They're just blasting the stuff off. These are lichens that are just having their life here. There's these indolithic caloplacas. You can see some apothecia really tiny in there in that hand lens. Lots of physia growing right on the building. Then on the benches, there's also lots of physias, xanthorias, and candelarias, and even a flavoparmelia. I've talked a lot, and I'll end things now. I like everybody for tuning in. And if anybody has any questions, I'm happy to answer them.
Great presentation.
Thank you.
How long did you stay in Madagascar again?
I was there for a little over 6 weeks. It was a long trip.
Yeah, 6 weeks long.
The longest so far.
It's wonderful.
Yeah, it was a great experience.
Hey, Todd, this is Greg. So are there a lot of these lichens that have that disjunct distribution between Australasia and Southern South America? Because, you know, in Basidiomycetes, there's some polypores, but in agarics, there's very few that show that distribution. So we assume the spores are getting around, but they don't become established. So I just was wondering what's going on, whether that was just a unique, you know, a rare event, or is that common?
You know, I think it's more rare than it is common. So, and I say that because the two other species that are really closely related to glabra are only found, well, there's Pseudocyphalaria homeophylla. It's only found in New Zealand. It's the sister species to glabra. And then Pseudocyphalaria freycinettii, which is the next closely related species, is only found in Chile. So glabra just has this ability to be able to, they say that it has the largest ecological amplitude of all Pseudocyphalaria species in the area. So it's able to just live in all different parts. And maybe that's one reason why it is successful at establishing there. It'll be really interesting to see what the symbiont patterns of genetic diversity are too, to see if it's associating with different, if it has this ability to associate with multiple types of algae. Or if it's, because you would imagine that if it's really specific and it can only associate with one group, then you wouldn't imagine it to be able to have such a distribution.
Very cool, thanks.
Susan Kaiser speaking. I was wondering if you could just go over your conclusion about how you got the diversity in the Southern Hemisphere bit. What role does evolution play versus transportation by birds or vegetation or what have you? Could you review that again?
So what I am still trying to figure out, but one thing that I can say is that dispersal is the main force driving this distribution. Mostly because our evidence is pointing at that the species is very young and it evolved relatively recently when the continents are already split apart. So long distance dispersal is the major force that is causing this distribution. And now the next thing to tease apart is what are the major vectors and what are the major directions of dispersal?
Thanks, that clarifies it.
I might be going in reverse of what they've been coming in, but I see one from Robert Salm. Todd, understanding that the nomenclature of lichens is based on the fungal mycobiont, has there ever been a pursuit to include the algal or bacterial symbiont in some way? There hasn't really that I've known. So when we describe a lichen species, we basically are only talking about the fungus, and the algae and other symbionts are identified. Like if I needed to identify the algae, I would have to use DNA to do that. where like the actual lichen I can identify using more traditional techniques. From William Carroll, how does lichens fit on the tree of life? On the fungal branch? Yes. Not on the, and just this kind of goes to the same thing where lichen taxonomy is all based in the fungal tree of life. And the symbionts are, if they're algae, they're in the plant tree of life. If they're cyanobacteria, they're in the bacterial tree of life. So we sort them out. And you know, lichens were for a long time considered plants. And so for a very long time, they were misclassified. And it wasn't until relatively recently, within the past, I think it was in the 1950s, when they actually considered lichens fungi. From Finn to everyone, are the 600 specimens from Madagascar 600 different species? If not, how do you decide? I think there's definitely not 600 different species. I definitely collected some duplicates of the same species. And I'm still working on identifying those. There's no identification keys to Madagascar. It's even hard to know what I'm looking at. Is it a new species, or is it something that we're just using a book that a key from Europe, and I'm identifying it as a European species? So there's a lot of work to do there, and that's going to be kind of the next focus of more research questions in the future.
Are the lichens, how well are the lichens known for South Africa in general?
I think a little bit better, but I'm not really sure. The flora of South Africa, so there's not a flora to South Africa, but there is one to East Africa. So that's been the key I've been using, but it's very out of date. And you know, it was written about 30 years ago. It's challenging to work on Madagascar lichens.
You'll have to write the next book.
Yeah, but I think I need to keep going back. I have a lot of work to do, a lot of learning to do.
And all of that rice to eat.
Yeah. Fortunately, I eat a lot of rice at home anyway, so that wasn't a big shock to me. I enjoyed it.
Oh, then there was a question about taking a group field trip to visit the lab at the Field Museum, which I guess is something to do, but on a future date.
I think that would be a great experience once things come back to normal. I would be really happy to give a tour of all the spaces of the museum and show you the fungal collection and our labs. That's a good idea.
Yeah, there's some of the labs were being remodeled and updated, and that got put on hold.
Mm-hmm. Yeah, like I was in the middle of I was on a roll working on Madagascar lichens, like every day. I just sitting down, identifying, and then the shutdown happened, and I had to basically just focus on all computer work. So I've been writing grants, writing papers, and that over, now it's been almost
10
90
weeks.
days. Yeah, it's like kind of driving me crazy.
It's even gotten to the point where learning a new skill isn't so interesting anymore.
Right.
There is another question. How difficult was it? I hope I'm not repeating by accident. How difficult was it to get the collection permits for each country location? What kind of paperwork is needed to import them back? Does the field have a team of lawyers to help you with the red tape?
I wish. It's really challenging. It depends on each country. So Australia, I collected in 3 different states, in Tasmania, in Victoria, and in New South Wales. And I had to get a permit for each one. And the whole process was different. Some of them you had to pay to get the permit. And then with New Zealand, it was basically like writing a grant proposal. And it took over 7 months to get it approved. And we actually had to cancel our trip that was planned for I think it was in May or March. And then we were planning to go in March and we had to postpone it to November to go.
For the paperwork to catch up?
Yeah. And it took like talking to people there, contacting people and getting them to ask questions about it. That was the worst. Chile was completely taken care of by somebody else who's from there. And that made it smooth. And Madagascar was challenging, even though I thought everything was, Steve he took care of everything, but still, I had to go physically take my specimens to a government office and show them what I collected. And then they would have to go to the typewriter and write up the entire document. And I have to wait for the typewriter to stop. And then, oh, is it ready? And then, so I spent a lot of time just sitting, waiting in government offices in Madagascar to get the export permit to take things out. And getting them into the country was even a challenge because then
Right.
U.S. Customs grabbed the specimens and took them from me, and I had to wait for a couple weeks before I got them back. So I'm lucky they didn't destroy them.
Was it Customs or was it the Department of Agriculture?
It was
people with the beagle.
Yeah, Department of Agriculture. They took it, and then I had to tell them, like, this is how I dry them out. I sterilize them with like bug spray. I try to do everything. And then after, I guess they were convinced that I took all the proper measures to not bring any pests into the country.
Well, dried mushrooms you're supposed to be able to bring into the country, which effectively is what you have.
Yeah, but the big thing was that I was collecting a lot of crustose lichens that are on bark. And so with crustose lichens, you have to take the substrate. So I had all these chunks of bark.
Oh.
And so the person that with the Department of Agriculture was like, no, I understand that lichens I had a letter from USDA saying lichens are exempt. You don't need a permit to bring them in. But then the person was like, yeah, but they're on plants, which are, and so I have to hold these. And so, yeah, that was a very scary time. That would have been a disaster if they just destroyed everything. Fortunately, they didn't.
Fortunately, it wasn't sausage. Then you really would not have too much leg to stand on. There's some more questions. Are there lichens that are toxic or beneficial to humans or wildlife?
So there are some lichens that can cause problems for, just like when I was in my master's program in Omaha, Bob Egan, my advisor, he got all these samples from Wyoming that were taken out of the stomachs of elk. And so they actually found a whole field of elk that died. And the one common thing that they had was a bunch of lichens in their stomach. and this is the genus Xanthoparmelia, which has a compound called usnic acid. And usnic acid is known to be antimicrobial. And we still don't really know if it's because they got too much. They might have been starving and they were just, the lichens were the only thing to eat. So they were eating that and then they might have concentrated it in their bloodstream and died. We don't really know for sure, but large elk that have a belly full of lichens, they all died. So I think there is, if you eat too many lichens, it could be toxic. But if you go to China, people use lichens for all kinds of medicinal purposes. There's a book on medicinal lichens of China. It's like this thick, and it has a picture and it says, "Oh, grind this up and make a tea with it, and it'll help your stomach," or and so on. So there's a lot of medicinal uses of lichens, but there's also some evidence that maybe they can be toxic, and they definitely are antimicrobial.
There's another question. Is there a difference in the permit needed between collecting fungi versus lichens from national or state parks?
I'm not exactly sure. I know that if you go into certain state parks, or especially national parks, like I wouldn't go collecting without contacting people and getting permission, because it would just be a big waste of time to go there and try to collect and then get caught and have everything taken from you. But I'm not really sure if there's a big difference between like collecting mushrooms and collecting lichens. It might be, maybe, I don't really know for sure.
Well, I think maybe, okay. Patrick, do you want to state what you just stated in the?
Finn asked if there's any delicious lichens. And I wrote that explorers reported that eating shoe leather was better than eating rock tripe lichen.
Yeah.
I heard that from, I think, Matt Nelson or someone, I forget who.
Yeah, it's probably true. Yeah. I still haven't tried to eat any yet. We do have some that they actually use these umbilicarias, this rock tripe lichen, in soups in Japan.
You can find it dried in the Asian market.
Yeah, and in India they actually use lichens and bryophyte, kind of like a mix of whatever they can scrape off the tree as a sort of spice for the food. So there are lichen. I think it's more used as a spice or some like a tonic to help cure things, not really to eat. Like they can't be. I don't think a lichen's ever going to be as delicious as a good mushroom, edible mushroom.
You just never know.
Yeah, at least there's fewer deadly lichens.
Yes.
Have you gone to any of the apothecaries in Chinatown? That might be interesting for you to look over sometime.
Yeah, I mean, my wife's from China, so I go to Chinatown a lot, and I don't know. We haven't really seen anything about lichens in Chinatown, but I think in a lot of the lichen use where they're collected and sold in China is really focused heavily in Yunnan province. But I still don't know enough to, and I haven't ever seen anything in Chinatown.
Okay, now somebody asked if you could show a slide the one showing the evolutionary species differentiation. I don't know if they're still here. I hope they are. I don't know if that's a pain in the neck to do.
Yeah, yeah, I can, let me pull that up again here.
If she's still here, she can, you know, unmute herself or unmute themselves and talk about what they were interested in. I hope they're still here. Well, maybe they moved on. Who knows? Sorry about that.
Yeah, that's all right.
Evolutionary species differentiation. I'm not sure which one that was. I don't know if she meant the tree, they meant the tree of life or one of your later things.
Yeah.
I don't think they're still on the Zoom meeting anymore.
Well,
Hi, can you talk about how closely there are other fungus and algae and cyanobacteria that are not symbionts? Like how closely are other like species related to lichens out there that aren't actually lichens?
Sure, yeah. So the lichenized fungi, most species fall into one group called Lichenoromycetes. But there's other independent origins of lichenized fungi. So there can be non-lichenized fungi that are more closely related to lichens than they are to other fungi. I always compare it to like the tree growth form in plants. It's not that all trees are closely related to each other. There's trees in many different families, and there's forbs in many different families. And so lichens are, there's been multiple independent origins of lichens. And there's even basidiomycete lichens too. So there's this capacity in fungi to associate with photosynthetic organisms. And we also see this, you could also think about like mycorrhizae are fungi that associate with photosynthetic organisms too. They are associating with the tree roots, and there's an exchange going on there, a mutualism. So that's a really, really interesting question, and it's really complex too. There's a lot going on. But I don't know, did I answer the question?
Yeah, pretty well. Thank you.
Yeah.
Well, part of the question I think was, how are the algae and bacteria not in the cyanobacteria not in lichens related to the ones that are in lichens?
Oh, yeah. So there's also on the other end of things. So we do find that the algae and are found in lichens, and sometimes can be found outside of the lichen and living independently. But most algae that are living in lichens are still really only known from lichens. And we don't know if that's because there's just not been enough sampling of like just on scraping a rock or pulling off tree bark to see like, oh, is this algae here too? So there's a lot of unknowns. And with cyanobacteria, we find that the lichens tend to pick them up from the substrate. So you'll have a community of cyanobacterial lichens that are growing on tree bark, and they all are sharing the same cyanobacteria. And then there's then you might find a bunch of them really close by, but on the soil. and they all have a different cyanobacteria, but they're all sharing, but the lichens growing on the soil are all sharing the same soil cyanobacteria. So they're able to pick this cyanobacteria up from the environment. The algae is a little bit different. It might be the same thing, but I think there's still a lot of work to do there.
Perhaps our last question of the evening. Can the same species of fungi associate with different algae or bacteria to form different lichen types? Are those classified as different lichen species despite having the same fungal partner? It gets complicated.
That's actually it brings up a really cool ability of some species of lichen. So there's a lichen that occurs on the west coast of North America called Ramelinum inzizii. And they showed that it's the same species of fungus, but it occurs in all these different ecosystems. In the south, in Baja, California, it's in desert, and it occurs all the way through California, Oregon, Washington, and all the way up to Alaska in temperate rainforests. And what they find is that the fungus is associating with different lineages of algae based on the ecosystem. So when the fungus produces a spore and the spore is brought to a different ecosystem, the fungus picks up the new algae that's appropriate for that ecosystem and can live with that. So they're actually able to switch. It's still considered Ramelinum inzizii, but it's able to live in all different types of ecosystems and associate with different algae. And then it's the algae that are different and the fungus that's the same.
Well, that's cool. Thank you so much for spending your evening with us tonight.
Well,
and we hope to meet you in real life someday.
Yeah, I'll be happy to come back and present again.
Great. And if you want to just come to one of our meetings or a foray, you're totally welcome.
Definitely. I really want to come to the forays too. And that will be a really great time.
It certainly will be. And of course, you know, anytime you're welcome at a meeting, though, goodness knows if we'll ever meet in real life this year.
Yeah, I hope so.
God, it's starting to get a little wearing, you know?
Yeah. This is real life.
Well, yeah, it is. It is. Yeah, you're right, Pat. You're right. Sorry.
But everybody looks a bit flat.
I believe that's true too. Oh, Lordy. Oh, all right. Well, good night, everybody.
Good night.
Bye.
Bye, and thanks again for a wonderful talk.
Absolutely.
Thank you.
Thank you.
We couldn't have done it without you.